All right. So, where were we? We've talked about cell division. We've talked about linkage, the discovery that genes are physically located on chromosomes. And we've worked out that chromosomes are made of protein and this stuff called DNA. And DNA turns out to be what Frederick Griffiths called the transforming principle. DNA from one type of organism, bacteria in his case, that passes into another type can actually change its phenotype. So we've got pretty strong case built up that DNA is physically responsible for heredity. So what exactly is DNA? We will step back in time to the early 1950s with a British scientist named Francis Crick, which rhymes with prick, over on the left, and a visiting American colleague by the name of James Watson on the right, Watson with the stupid hair, studying DNA in England at Cambridge University in the lab of senior scientist Maurice Wilkins. Also working in the lab with them is Rosalind Franklin, and I might add, I feel chuffed that I can finally talk about a female scientist because she was a good one. She was one of the world's experts in a technique called X-ray crystallography. How this works, okay, when I was a kid, my grandmother used to like having, ornaments and little stained glass bits and pieces in her windows. And she had a sun catcher that's a big chunk of very clear glass that's made in the shape of a crystal. And every time a sunbeam would hit that sun catcher, the beam would get diffracted, meaning it got broken up into a bunch of beams. And what we'd get would be a bunch of spots of light on the wall. She also had a crystal chandelier in the dining room, and that had lots of little glass crystals hanging from it. And the light from the chandelier would hit these crystals, and every beam of light hitting a crystal would be broken up into multiple beams of light, and you'd see a whole bunch of spots of light. And I can remember she'd reach up and spin the chandelier, and all of those spots of light would go round and around on the ceiling. And when I was 2, I thought this was the coolest thing ever. But then when you're 2, everything is the coolest thing ever. So I'm assuming some of you have seen this. If you hold a crystal up to the light, it will take a beam of light and break it up into little spots of light. We call that diffraction. And what Roz Franklin was so good at was much the same thing, except with X-rays. You take a substance, some kind of material that you're studying, you make a crystal out of it, and you shine, instead of regular light, you shine a focused beam of X-rays at it. Well, that crystal does diffraction, and it breaks up the X-ray beam into many beams. And those many beams passed onto a sheet of photographic film, which you then, I don't know if any of you remember film. It's what used to be in cameras before we all just got camera phones. But that film can then be developed, and everywhere a beam of X-rays has hit the film, you get a dark spot. Just like if you're looking at a glass crystal in a beam of light, you get light-colored spots on the wall where each beam of light is hitting the wall. Here you get dark spots wherever a diffracted X-ray beam has hit the film. And it looks like this. You can see that there is, some complexity going on. Those dots are not just arranged in some kind of random order. And here's the thing, if you measure the position of the dots on the exposed film and you do some seriously gnarly math, you can work out how the atoms in the crystal are arranged. right? We still haven't gotten to where we can see atoms, but you can work out the spaces between atoms that make up that crystal by measuring the positions and the distances between all of those dots. And this way you can work out the shape of the molecule that you're working with. So even though we cannot see actual molecules because they're far too small, even with powerful light microscopy, we can learn about how the atoms that make them up are arranged. Okay, so we've gotten a little bit deeper into that black box. Now, this is very difficult to do, and Franklin did not invent the technique. That was invented before her time, but she was one of the best in the world at it. And she was able to show that the DNA molecule was shaped like a coil. Here she is on the left. On the right, that is one of the most famous pictures in the history of science. That's what she got when she made a little crystal out of DNA and hit it with a beam of X-rays. That's the pattern of black spots that she got. And, you know, I can't personally make much sense of it because I'm not trained in X-ray crystallography, but that particular pattern, especially that pattern of dots in an X shape, is typical of molecules that are shaped like coils, or as we say, that they are shaped like a helix. And a helix is any coil or any spiral. A bed spring is a helix. A spiral staircase is a helix. A snail shell is a helix. A corkscrew is a helix. And the DNA molecule must be shaped like a helix. We know it's a polymer, so we know it's a bunch of subunits that are strung together in a long chain, but the chain must have some sort of spiral twist to it. Maybe it looks something like a rope or something like that. Ross Franklin was able to work out just from pictures like this, not only that the DNA molecule was a helix, but she was able to work out about how wide it was and how tightly the coils were wound, what the space was between one coil and the next coil. You know, she was able to work out here's a helix for you. Sorry, Roz. And she was able to work out not just the width of the helix, but how much distance there was between adjacent coils. So she worked out the proportions and the shape of the DNA molecule. Watson and Crick got hold of Franklin's data, be it said they got it without her knowledge or permission, which pardon me saying this, but I don't really have any other way of putting it. Taking somebody else's data without their knowledge is, my apologies, kind of a dick move, right? There, no other word in the language adequately expresses it. So they knew that the DNA molecule was a helix. They knew how wide and how tight the helix was. They also worked out that the phosphate groups on the nucleotides had to be on the outside. They also knew how large nucleotides were. They knew the shapes of nucleotides, and they knew about Chargaff's rules, and they'd actually learned them from Erwin Chargaff himself in 1952. So they basically locked themselves in the room and tried to build models of DNA that would fit all of these criteria. You know, knowing what the parts of DNA are, knowing the shape of the parts, how do we fit them together in such a way that that our fitting together will match the shape of the entire thing? April 25th, 1953, they published a paper describing their model for the shape of DNA. And there's Watson on the left and Crick pointing to it on the right. That's the model that they built. And DNA turned out to be not just a single polymer of nucleotides, but two polymers wrapped around each other, forming a structure that's not just a helix, it's a double helix. Two spirals spiraling around each other. That's a slightly easier picture to see than the old photograph that you saw on the previous slide. On the left, that is a computer reconstruction of a DNA as what we call a stick model, where the bonds between atoms are just represented by little sticks. On the in the center of the slide is exactly the same DNA molecule. This is just represented by, here we make the atoms look like little ping pong balls, and the different different elements are shown by different colors. These are simply two ways of showing the same thing. And in both of these, the sugars are shown in pink and the phosphates are shown in white. And if you look at this, like right over here on the left, this thing that I'm highlighting in yellow is one string of sugars and phosphates. That's one of the strands in the helix. And it is wrapped around the other strand, which I'm highlighting here in green. And this strand is also made of the sugars and phosphates. See, they're on the outside. So the two strands, the backbones are made of sugar linked to phosphate, linked to sugar to the phosphate. And notice that the bases, which are shown in red and yellow, project into the interior of the helix. So there's a base, 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 there's a base. And they link up with bases over on the other strand. There's a base, 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 there's a base. And yeah, the bases are really important. So these are just two different ways of showing the same molecule. And I put them up there so you can see what we mean by a double helix. If you could untwist a piece of DNA, it would actually look like a rope ladder. And over here on the left and the right, I've represented each phosphate by a little tan ball and each sugar as a green pentagon. And so that's not showing up really well. Let me give it a different color. Okay, all of this is a sugar linked to a phosphate, linked to a sugar, linked to a phosphate, linked to a sugar, linked to a phosphate in a long chain. And so is this. There's a sugar linked to a phosphate, sugar linked to phosphate, sugar linked to phosphate, sugar linked to phosphate. The sugars and phosphates bond to each other in these long chains, and they form the sides of the DNA ladder. Whereas the bases, which, let me highlight those in blue, I guess, extend inwards. Like there's a guanine, there's a guanine, there's a guanine, there's an adenine, there's a thymine, and there are the bases on the other side, and they form what we might call the rungs of the ladder. So the bases make that rungs of this rope ladder shape, and the sugars and the phosphates make up the sides of this rope ladder. And then the entire thing twists. So when it's in its natural position, it has that helix shape, like a twisted rope ladder. So that's another computer graphic of that ladder twisting into a spiral to give us that double helix shape. Incidentally, Watson and Crick were working this out, but they couldn't actually see what they were doing because molecules are far too small to pick out with ordinary microscopy. In the past 20 years or so, however, we've developed a technique called atomic force microscopy. I don't need to go into details of how it works. And this is a picture of an actual DNA molecule. Up at the top, that's what one looks like using this technique. On the bottom, labeled B, that's the same molecule, but computer correction has been applied to the image to make it look straight. And you can actually see the little spiral grooves that it's got. That is a single DNA molecule, and it's got the spiral shape. That's a double helix. You can compare the graphic up here, that part, with this bit down here. That's a computer graphic of what a DNA molecule should look like. And you can see that that model that Watson and Crick worked out down on the bottom is pretty darn close to what an actual DNA molecule does look like using atomic force microscopy. We've got a technique now called scanning tunneling microscopy that gives us even more magnification. There's a model on the left. On the right, that's an image of actual DNA taken with scanning tunneling microscopy. And Watson and Crick basically got everything right. And you should be able to see, let me highlight it for you. I'll do this in red, right? That's one of the backbones right there. And that's another one of the backbones coming around right here. And then you can even see some of the bases that are in between them. There's another piece of that backbone, and there's some more of those bases there and there and there. And there's some bases kind of dimly visible there. So Watson and Crick basically got it right. And we can confirm that with much more sophisticated microscopy techniques than we had back then. Little bit of human interest stuff before we go on to the significance of like why anybody should care that the DNA molecule looks like a double helix. Watson and Crick and their supervisor, Maurice Wilkins, won the Nobel Prize for Medicine for this discovery in 1962. Roz Franklin probably has not been treated fairly. People have argued that she should have shared the prize because her data was absolutely crucial for what Watson and Crick did. Unfortunately, she died in 1958. She died young, and the Nobel is not given posthumously. You can't win a Nobel Prize after your death. It's also not shared by more than 3 people. That's just the rules of the contest. So technically, she could not have shared the prize. That being said, her importance has often been overlooked, and that's in part deliberate. Watson published a book in 1968 called The Double Helix, and it's a really good read because it gives you, a ringside seat into the mind of an extremely intelligent scientist. Watson is really smart. You can't do what he did without brains. And it gives you a good idea of what it's like to be a really smart scientist working at the cutting edge, solving a really difficult problem. I mean, he takes you there like few people can. Unfortunately, Watson is a great scientist, but he's also kind of a bro. There's no other way to put it. And when he wrote about Roz Franklin, he mostly complained about how she didn't dress nice. made her into this kind of, cold, boring, dowdy bitch. As he wrote, she was not unattractive and might have been quite stunning had she taken even a mild interest in clothes. This she did not. There was never lipstick to contrast with her straight black hair, while at the age of 31, her dresses showed all the imagination of English blue-stocking adolescence. Right, so she's a scientist and he's mostly complaining about how she dresses. Like, who cares? Here's the thing. It's not even true. Rose Franklin never did marry, but she did have lots of friends. She could be a very fun-loving person. She loved going to the theater. She was a perfectly lovely individual. At work, though, she was all business. She did not fool around. She did not dress to impress. She had a job to do, and she did it. She was very professional. And I think she fell victim to the kind of double standard that women tend to fall into in the professions, where if you try to be sweet and nice and kind to everybody, you get walked over. And if you try to be cool and professional and get the job done, you're likely to get, called a bitch behind your back. And you're going to have people saying, you'd be prettier if you smiled more. And that's a problem that women face in the workplace. And hopefully it's not as bad as it used to be, but it's still definitely there. That if you try to be nice, you're not taken seriously. And if you try to be firm and businesslike, you're seen as, cold and unfriendly. You can't win for losing. And Franklin definitely fell afoul of that. She was a lovely person, but she was a very serious in the workplace, and that got her maligned by James Watson as, this very drab and unfashionable person who really wasn't all that important. She does deserve a large share of the credit, even if she couldn't win the Nobel, because her data were the key to solving the puzzle. And it's worth noting that when he published a revised edition of the double helix, Watson did later acknowledge how important she was, writing that we both came to appreciate her personal honesty and generosity, realizing years too late the struggles that the intelligent woman faces to be accepted by a scientific world, which often regards women as mere diversions from serious thinking. So she kind of fell victim to the boys' club here, but even if it's a bit too late for her personally, her importance is recognized more. And Watson and Crick have both basically admitted that she deserves a large share of the credit for the discovery that they made. Now, if we could just get Watson to shut up about race, that would be even better. He is still around. He's quite old. He's gotten into hot water several times for having spouted off about Black people being less intelligent than whites because of their genetics. Yeah, he's a racist jerk. And I'm not teaching you about any of these scientists in the past because I want you to look up to them as heroes. They're people. They all have their flaws. They all have their biases, some of them more obvious than others. They've all got feet of clay in some regard. And I'm not teaching you about them because I want you to revere them as, great heroes of the past. They're people, and science is done by people. Sometimes it's done by people that hold rather objectionable views. Just as great music is sometimes made by people who are absolute assholes. It's just the way people are. So I'm not teaching about these people because I want you to look up to them as role models or accept them as, saints or magnificent human beings, you know, I don't really care that much about the people at all, but I want you to understand the processes that give rise to scientific discoveries and the humans process by which those discoveries get made. So yeah, Watson is still kind of a racist jerk. But he did discover this. Watson and Crick, with Ross Franklin scoring a major assist, worked out that DNA is shaped like a double helix. Who cares? Is this more trivia that you're just going to have to cram into your head just long enough to spit it back out on the exam? Here's the thing, and this is going to be the theme for the next couple of weeks. If you want to understand how something works, you have to understand its shape. If you want to understand, how a screwdriver works, you have to understand its shape, right? There's a Phillips head screw on the left, and there's a Phillips screwdriver on the right. And the shape of that screw head and the corresponding screwdriver, those shapes are important because they determine what they do, right? The shape of the screw, right? The threads and the lag, if there is one, and the shape of the grooves in the head determine what you can do with the screw and what you can use it for. The shape of the screwdriver determines what types of screws it can drive. And the same is true for hammers or saws or any tools you care to mention. They're made to particular shapes, and that shape is the key to what they can do and also what they can't do. The shape of that screwdriver head is critical to understanding what types of screws it can drive and what it can't drive. Like it can't drive slot-headed screws or hex screws. Shape is what determines function. And the same is true for DNA. Understanding DNA's shape is not just some rando exercise. It's the key to understanding what DNA actually does and how it does it. So if we go back, DNA is, again, a polymer of subunits. The subunits of a polymer are called monomers, and the subunits of DNA are called nucleotides. And to be a little more accurate, DNA is not just one polymer, it's two polymers wrapped around each other. Remember that double helix shape. And remember, every nucleotide consists of an atom of the element phosphorus surrounded by 4 oxygens. That's the phosphate. And the phosphate is connected to, highlighted in green, a molecule of a simple sugar. That's a ring of carbons, hydrogens, and oxygens that in this case is a type of sugar called deoxyribose. And the deoxyribose sugar is compared to one of 4 single or double ring compounds containing both carbon and nitrogen called the base. And whereas every nucleotide has identical sugars and phosphates, there are 4 bases that are used in DNA. Adenine and guanine, shown in red and green, those are both double rings of carbon and nitrogen atoms. And thymine and cytosine, which are both single rings of carbon and nitrogen atoms. Okay, and DNA is a polymer of those, or a double polymer. And again, what holds a DNA molecule together are you have bonds between the phosphate or the sugar of one nucleotide and the phosphate of the next nucleotide, and those sugar to phosphate to 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 sugar. That's the backbone of DNA. That is what holds those 2 strands together. And here you've got it on the other side, phosphate to sugar to phosphate to sugar to phosphate to sugar to phosphate to sugar, phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. Those make up the sides of our ladder. The bases of each strand project towards each other, and they are held together by easily broken weak bonds called hydrogen bonds. So these things that I've represented by dashes right there. And the thing about hydrogen bonds, we don't need to know, there's several different types of chemical bond, ways that atoms stick together to make a molecule. We don't need to go into ionic, covalent, and metallic bonds. We've got a perfectly good chemistry department if you ever need to know that. All I'll say for our purposes here is hydrogen bonds are relatively weak, and they're easy to form, and they're easy to break. You could think of DNA as being kind of like a twisted zipper. The sides of a zipper are built very tough, right? It's very difficult to cut or tear across a zipper, but the little bonds that the zipper's teeth make are easy to lock together as you zip your zipper, and easy to break apart as you unzip your zipper. In fact, that's the whole point of having a zipper, that the bonds that hold a zipper together are easy to zip and easy to unzip, right? That's why we use zippers in the first place. And so you could think of DNA as kind of being like a twisted zipper, with the bonds between the teeth of the zipper that easily form and easily break being analogous to the hydrogen bonds between bases in a DNA molecule. And thanks to the way the bases are shaped, if you have an adenine on one strand, it can only form hydrogen bonds if there's a T on the opposite strand, and a T can only form hydrogen bonds with an A. The red dashes here show you what those hydrogen bonds look like. Again, we don't really need to worry how hydrogen bonds work. Just know that they are weak bonds that are easy to form and break up. And an adenine can only hydrogen bond with a thymine on the opposite strand. And a guanine on one strand can only form hydrogen bonds with a cytosine on the other strand. We call this base pair complementarity. An A on one strand can only bond with a T, and a G can only bond with a C on the other strand. And this is what explains Chargaff's rules. Remember, Chargaff is the guy who worked out that the percentage of A in a DNA molecule was always equal to the percentage of T, and the percentage of G was always equal to the percentage of C. And that is because every A on one strand must have a T on the other strand, or those hydrogen bonds will not form properly. If there's an A on one strand that doesn't match with the T on the other strand, you've got a busted DNA molecule, kind of like a zipper where just one of the teeth is bent out of shape or moved out of place a little bit. You ever had a zipper that's always getting stuck because one tooth is broken? That's what you would have if you didn't have a T for every A and a G for every C. You wouldn't get the double helix forming properly. So you can count if you want to. There's exactly 6 guanine bases, 6 Gs on this diagram that I wrote, and every G's got a C. So there's exactly the same number of cytosines as there are guanines. And there's exactly the same number of adenines, 2, as there are thymines, also 2. That explains Chargaff's rules. One of the annoyingly fun questions I like to give on exams is I'll give you some DNA nucleotides. Let's say we have a DNA sequence on one strand that is a G, an A, a T, a T, an A, a C, and an A. It was actually the title of a movie. If one strand of DNA spells out GATTACA, what must the other strand be? Well, if one strand has a G, the other strand has to have a C to match with it. Where one strand has an A, the other strand has to have a T to match up with it. Where one strand is T, the other strand has to have an A. And where one strand has C, the other strand must have a G to match up with it. So if one strand spells out Gattaca, Gattaca, the other matching strand must spell out CTAATGT. That's your explanation for Chargaff's rules. A DNA molecule will not form properly unless the bases match up in this very precise way. And it turns out that base pair complementarity is also the key to DNA replication. DNA is present in the nucleus of every cell, and for a cell to divide, DNA has to duplicate. DNA has to be copied. And when a cell duplicates a piece of DNA, first of all, the hydrogen bonds that lock those two strands together break, and the two unzipped strands move apart. Now, who is doing the breaking and unzipping, I'll get to later, but we don't need to worry about who's doing it right at this moment. As far as you're concerned, it's leprechauns that are unzipping the DNA and moving the strands apart. It's not really leprechauns, but we'll talk later as to what really is making the move. And once the unzipped strands have moved apart, every strand can serve as the template for building a complementary strand. So if there's a G here, we have to fit in a C next to it. And if there's an A on this strand, we have to fit in a T next to it. And next to the T, we have to fit an A. And every C must have its G. So we've made a new strand that's complementary to the old one. And looky, our new strand has exactly the same order of nucleotides as the old strand over on the far right. And then this old strand can also be used as a pattern to assemble new nucleotides. So we have to bring in a G here. And again, exactly who is bringing in the new nucleotides and welding them together, I'm leaving aside for right now. We'll get to later who's doing that. If you want to think of it as little leprechauns that are moving everything around, that's, well, that's silly, but we'll get to something better later. So there is our new complementary strand right there. And you'll notice that it is exactly identical to the one over on the far left. So the two unzipped strands move apart. And then each old strand is used as a pattern to assemble a new strand. Look. See, it's computer animation. So our end result is we now have two identical strands of DNA, because two identical pieces of DNA where each of those separated strands was used as a pattern to build a new strand. The technical name for this is semi-conservative replication. And this is key to the way that we can take any piece of DNA and duplicate it very, very exactly, because we can use each strand of DNA as a pattern for making a complementary strand. So each strand of a DNA molecule is used as a pattern or a template to make the complementary strand. When DNA replicates, the hydrogen bonds unzip, and then each of the separate, that's what's going on right here. Those hydrogen bonds are unzipping, and then each strand becomes the pattern for assembling a new strand. And yes, it happens in opposite directions on different strands. We don't need to get into that now. And the replication's being done by a couple of enzymes called DNA polymerase and DNA ligase. Those are the little leprechauns that are doing the work. And I'll explain later what they really do and why they're not actually leprechauns. We now know that each chromosome is fundamentally a single long piece of DNA. In fact, if we could take all of the chromosomes out of one of your cells and somehow unravel them all and put them together head to tail, we'd have about 6 feet of DNA. That's roughly how much DNA is packaged in each one of your cells. Now, when a cell is in interphase, that DNA is just lying together in a lump, kind of like a very large plate of spaghetti, right? If I brought you a humongous platter of spaghetti, you wouldn't be able to count the noodles because the noodles are all tangled up with each other. Well, that's kind of like what a cell nucleus is like when the cell is not about to divide. The DNA is there. It's just not compact. But at cell division, what's really going on during prophase is that the DNA coils tightly on itself, and then the coils coil onto the coils, and then the coils of coils coil onto the coils, and then the coiled coils of coils coiled onto the coils. It's a process called supercoiling. And the DNA coils on itself and wraps around a core of special proteins that happen to be called histones. That's a photo of an actual human chromosome that is partly unwound. This was taken, this picture would have been taken during prophase. And each one of these chromatids, right, that's a chromatid. And this is a chromatid. And each one of those chromatids is just one long strand of DNA coiled in on itself, coiled in very compactly. And this is why you can't see chromosomes in interphase. They're there, but when they're unwound, you can't make out individual chromosomes. When prophase hits, the DNA is winding itself into these tight coils onto this protein core, and the chromosomes become compact and large and visible to light microscopy. That's when you can see them. That happens to be a chromosome that's been treated to unwind the DNA. This stuff down in here, those are the proteins that it was wrapped around. That's kind of like the old spool that a piece of thread might be wrapped around, all those proteins. And then surrounding it, you can even see individual strands right up here. If you look really carefully, that's all the DNA unwound off the histone proteins. And it looks like an extremely large platter of spaghetti. Only at the edges, kind of where I'm drawing arrows, can you even make out individual parts of the molecule at all. And all that gray shady mess is what used to be wrapped around that core of histones. And DNA replication is what happens during that mysterious S phase of the cell cycle. During that S phase, each chromosome replicates. Each chromosome is one long piece of DNA. Each chromosome replicates to form 2 copies, and each copy is one of these chromatids joined at the centromere. So each one of these chromatids is a nearly exact copy of the other. That's what's going on in the S phase. And I could go on, but it is 10:50. So I'm going to go ahead and stop the recording, and we will pick this up on Monday. Same bat time, same bat channel.